In this lecture, we are going to talk about the basic pharmacodynamic concepts that are needed to understand the relationship between drug concentrations and effect. Let's start with the definition of pharmacodynamics, which is the relationship between drug concentrations at the site of action and the resulting effect that we measure. Remember from the last lecture that drug concentrations in blood, plasma, or serum often serve as a proxy for actual drug concentrations at the site of action. So what we notice is that with increasing drug concentrations in either the plasma or at the site of action, there is an increase in effect, but this effect eventually plateaus at a maximum level, which we label as being the efficacy of the drug. We can plot drug concentration on a linear scale, as in the graph on the left-hand side, or on a logarithmic scale, as seen in the graph on the right-hand side. The advantage of converting drug concentrations to a logarithmic scale is that it amplifies the initial lag phase and linearizes the portion of the graph when effects increase exponentially with increasing drug concentrations. We can use concentration effect graphs to compare different drugs that have the same effect. In the top figure, we see two drugs that have the same maximal effect, but higher doses of drug B are needed than drug A to achieve the same effect. We therefore say that drug B is less potent than drug A. In the figure below, we see two drugs with different maximum effects. We say that drug B is less effective than drug A. Remember from the last lecture that, for most drugs, the intensity of effect is related to the drug concentration at the site of action. This is because an effect is brought about by an interaction between the drug and its receptors. So the higher the concentration of the drug at the site of action, the more drug molecules are available to interact with the receptors, the more receptors are occupied, and therefore the greater the effect. Note also that once all the receptors are occupied, an increase in drug concentration will probably not result in any greater an effect, and this causes the plateau that we saw in the concentration effect graphs previously. When we talk about a drug's effect, we do not just mean the intensity of the effect as discussed in the previous two slides, but we also refer to the time course of this effect, by which we mean how quickly the effect appears and for how long it is maintained. To study this, we would plot the effect versus time rather than dose. In addition to the concentration at the site of action, there are other factors that determine the intensity of the drug's effect, which include the density of receptors on the cell surface, because the greater the number of available receptors, the higher the likelihood for a drug-receptor interaction, and therefore the greater the response. Also, the greater the number of receptors, the higher the maximum effect will be. Second, in addition, the mechanism by which the signal is transmitted into the cell by second messengers can serve either to amplify or limit the effect of the drug-receptor interaction. And finally, regulatory factors that control gene translation and protein production can either dampen or amplify a drug's effect. We also need to explain the duration of the effect, which may be due to the time that a drug remains engaged with the receptor, but can also be modulated by intracellular signaling mechanisms or gene expression. The consequence of all these factors contributing to the effect of a drug is that, despite having similar drug concentrations in the blood or at the site of action, different individuals may vary in their sensitivity to the effect of a drug. In addition, these factors can help explain how animals can develop tolerance to a drug's effects. So let's conclude with a brief discussion of the development of tolerance to a drug's effects. Animals can develop tolerance either due to pharmacokinetic or pharmacodynamic factors. Pharmacokinetic factors would include an increase in drug metabolism leading to a decrease in drug concentrations for a given dose, whereas pharmacodynamic factors refer to effects relating to how the presence of the drug can alter the expression of either the receptor or its second messengers. In this final slide, we see that when tolerance develops, we need higher and higher doses to get the same effect. But if this tolerance is due to pharmacokinetic factors, those doses are actually resulting in the same concentration in the plasma and at the site of action, which is why we are seeing the same effects. However, if the tolerance is due to pharmacodynamic factors, what we see is that higher concentrations at the site of action are actually needed to elicit the same effect.